Thank you so much. The honor is given today to the Chemical Genomic Core Facility to present one of the 4 services. I have to stress that we have the phage display service under my control. We have the high throughput screening under the control of Jinwei Mi, and then the 600 NMR under the control of Dr. Atten. Today's talk is done by Dr. Li Fentong on the medicinal chemistry and informatics. So we're listening.
First of all, I would like to thank Joe from CTSI to organize the seminar for our core, and thanks Alain for your kind introduction, and thanks everyone to join the seminar today. So I'm basically working on or taking care of the medicinal and the analytical chemistry part in the chemical genomical IU School of Medicine here in Indianapolis. So first of all, I would like to give you a brief introduction of the aim of our core. So we provide shared scientific and technical resource to both IU School of Medicine and all the users from CTSI campus for their grant applications on drug discovery and the target validation. We also provide instrument usage and teach and train students, postdoc, and the technicians in associated research techniques. And we do support high impact publications, help our users to generate IP, and of course, we also contributed to the faculty and the student recruitment and the retention. So we're located in Medical Science Building, Indianapolis, and we have our website as well. So first, we have, as Alain mentioned, we have a variety of expertise and the surface on phage display, molecule design synthesis analysis, high super screening, and SAR by NMR. We also have advisory committee, which from different departments within IU School of Medicine, they represented for broad expertise and experience on drug discovery related area. So we served diversified users within IU School of Medicine, IUB, Purdue, and also Notre Dame. So over the past 5 years, we helped more than 70 research groups, and we support more than 100 grants. Of course, letter of support, publication, patents as well. So our users are from diversified department and campus. So today I'm mainly talk about the scope of the medicinal and the analytical chemistry capability in the core. So first of all, we participate in your grant and we provide a letter of support, cost estimation for your grant application. And since we have a variety of instruments for compound synthesis, purification, and analysis, we also provide a lab space and instrument usage and the trainings on students, postdoc, and technicians to use our instrument in the core. So besides the instrument usage, we helped our users on compound information retrieval and the compound recommendations, design, synthesis, and analysis. So first of all, I would give you an introduction of the synthesis capability in the core. We have like 5 chemical hoods to perform the reactions. Actually, so we have 3 8-fit and 2 6-fit hoods, and the hood space will be good for at least 5 chemists to work in the core at the same time. And we do have instrument to help the reaction, such as microwave reactor. It could control the heat and deliver the energy to your reaction system precisely. And it can control the temperature and the time of your reactions. We also have a parallel synthesizer, which enables all the users to perform multiple reactions at the same time. So this reactor is good for a regular reaction, also peptide synthesis. Of course, we have a bunch of instruments which used to purify compounds and peptides. So first of all, we have 2 waters preparative HPLC system, which is good for reverse phase purification. We have different size of columns, so we can purify from milligram to a few milligram to a few hundred milligram reaction mixture at the same time, at one single injection. And we also have 2 biotage flash purification systems to do normal phase purification. And we have load of vapor and multi-channel nitrogen dryer to dry out the fractions. So next will be the analytical instrument. So the major instrument in the core for the compound analysis is LC QTOF. So the QTOF is a high resolution tandem mass spec. It has a quadruple mass and a collision cell in between, and it has a TOF in the end of the instrument. And the LC is ultra high performance LC with quaternary pumps, and you can deliver 4 mobile phase at the same time. And the dye array detector we have can monitor up to 8 wavelengths at the same time. So it enables us to provide high accurate mass analysis up to 4 decimals for small molecule peptides and oligonucleotides. And since it's a high resolution instrument, so it's good for intact protein analysis, also modified protein analysis. So I will give you guys an example in the later slides. So since it's a tandem mass spec, we can do LC mass analysis in both targeted and untargeted modes. So the other analytical instruments include a Brook 600 NMR with cryo probe. Yeah, of course, we can use it for small molecule structure elucidation and verification, but we also have Dr. Bertrand Roy using this instrument working on SAR by NMR. So he's going to give a seminar, I think, early next year. And we have some like small instrument, but useful instrument for melting point detection and the polarimeters for chiral compound analysis. So next, I am going to focus on the service we will provide. So we have 200,000 compound libraries, so which is mainly used by Jingwei in the core to do high throughput screening. So what I do here is I can provide information management and retrieve the information and do compound recommendation and search the compound by similarity and the substructures for your assays. And we can also help you to evaluate the heat and the lead compound properties. So we have the Schrodinger's drug discovery platform right now, but and we are building. It's not we haven't put it into service right now, but we are going to put it as a service next year. So the synthesis. So we will help you to do the design, synthesis, and purification of small molecule as inhibitor probes and also activators. We also do provide peptide and modified peptide synthesis, as well as focus the compound library synthesis. So usually we work on milligram or gram level synthesis, but we can provide kilogram synthesis through contract research organization. So usually we will provide a purity over 95% by NMR and LC mass. You may want to know how it works. So first, you can send your request to me on compound structure, or if you are not very sure about the final compound you are going to design the synthesis, and feel free to talk to me and we can help you to decide the compound. So after that, you will get a quote from me with the price and the estimation of the time for material and the synthesis. And as an output of the project, you will get compounds over 95% pure, either purified by silica gel or HPLC. And you will get a LC mass and NMR spectrum as a proof of the structure and purity. So through the past few years, we're working on a variety of targets, and we have synthesized a variety of molecules, such as those inhibitors and other stabilizers, probes, activators, unnatural amino acid, peptides. So you might wondering the cost of the synthesis, also the purification of the proper HPLC. So I will just give you a rough idea of the cost. It's a case by case scenario because the cost of the starting material is always different. But I just want to give you a cost estimation. Like for 5 milligram of the two-step reactions for regular small molecules, it will be over $1,000. But for the peptide synthesis, it would be just a few $100, usually for like 20 amino acid peptides. And we also provide preparative HPLC surface. It's like a few steps. We have to first check your sample qualities before the prepper by the LC QTOF. Then we charged a few hours on method development on both instrument, labor, and material cost. And we will check all the fractions for you again, and dry out the fractions and check the fraction after dry out, make sure it's still good, and you can request for an NMR test as well. So the next major service we provide is the molecular analysis. As I mentioned before, we're using LC QTOF to do analysis on small molecule, peptides, oligonucleotides, proteins. So how it works, you can either submit your sample to me, or the users can get trained by me. So the cost will of course be determined by the labor cost, instrument cost, and the material cost. So how it works, usually the user will need to communicate with me on your analysis needs, such as your basic sample information and analyze type and the structure and the concentration of your analyze in your system. So basically we have two routes. Route one is after the discussion, if the users need to use the instrument in a daily base or in a very regular base, so we can set up the training for the users. usually we will set up an account from iLab because the users need to reserve we have a very good instrument reservation system from iLab. So you have to set up an account from iLab, and then you get trained by core personnel to use the instrument. So after that, you can reserve the instrument online. For either you can use it by yourself, or anytime you have problem, or you need some assay development, you can request for supervised usage. Or if you don't want to develop or test the sample by yourself, you can of course get a quote from me. And our core will do the compound analysis for you. So we provide help from sample preparation to data acquisition, spectral analysis, and the report for your grant application or publication. So I would like to talk a little about of sample requirement here as well. So usually you would need to use like LC mass grade solvents like water, acetonitrile, methanol, and you can include a small percentage of DMS in your sample. So the injection loop, our LC QTOF is 20 microliter. So you don't need more than 20 microliter sample. Just a little bit dead volume should be fine. So the most important thing is you have to filtrate your samples, at least centrifuge your samples. So for the LC mass analysis, usually we will start at 100 micromolar for small molecule. And for protein, for large molecule like protein, nucleotide, you can start at 10 micromolar. And for the untargeted and the targeted LC mass analysis is more sensitive. So usually, we can detect at the picomolar level, giving injection of 20 microliter. For like small molecule NMR tests, so a few grams usually would be good for proton and even the carbon NMR. And you can recycle your sample after the test. So in next few slides, I will give you a few examples of what the LC QTOF can do. So whenever you have an application, feel free to contact us. So the first example is to test the purity and the identity of the compound with a high resolution mass. So I'll take reserpine as an example here. It's a market drug. It has molecular weight of 609. So on the positive ionization mode, you should see a M/Z at 609.2807. And this is the experiment data. You see a molecular weight at 609.2816. So if you calculate the error by minus the calculated value from the observed value, you will see the error is very small at 0.0009 Dalton. So in terms of the ppm, error is 1.5 ppm. So our instrument can provide very accurate result for to support high profile journal. And we can also test the intact protein. For example, I have a protein APE1 with the average mass at 65,000 Dalton. So from the left, you can see a raw spectrum. So the protein will carry different number of the protons, and each peak here represent for a different number of the protons the protein carry. And here 922 0.0098 is the internal reference ion to calibrate the instrument, real-time calibrated instrument. And after the deconfusion, you can find the observed molecular weight at 35642. And the difference of the observed value and the calculated value is 1.1 Dalton, which is pretty good for the protein. So you can easily tell whether you have the right protein or not, or you can tell if you have some modification on your intact protein or not. So we have the users to use our instrument to do like covalent inhibitor discovery. You can very easily to tell whether your small molecules bind to protein or not. And from the deconfusion result, you can easily tell what's the percentage of your protein has been modified. Okay, usually, so in this slides, there's some information about the cost of the LC mass and NMR analysis. Usually, you can either choose to reserve the instrument and just pay the instrument rate, or you can submit the sample. This is more for the sample submission. So we provide help on sample preparation, data acquisition, and analysis. So if it's just regular sample and it didn't need too much sample assay development, the cost will be like within $100, including the data acquisition, data analysis, and a brief report. So my next example would be the compound water solubility. So why the drug water solubility is important? Because usually poor water solubility leads to low bioavailability, and it provides challenges with metabolism or permeability. So here's some definition of the water solubility from very soluble to insoluble. So basically, there are two types of water solubility. The first is named kinetic solubility, which is accessed by diluting a test compound in DMSO stock into a water buffer. And in this case, usually you keep the final DMSO concentration at 1%. And the second is called thermodynamic solubility. So it's just invested the solubility of your compound as a saturated solution. So you basically put your compound just in whatever buffer like you want to use, and then incubate it for overnight, and then you filtrate and then test the concentration by LC mass. So I'm going to talk a little about how to determine the two types of water solubility by LC mass, or sometimes you may need to develop LC mass assay. So first of all, of course, you need to do a condition optimization of the LC and also the mass detection assays to choose the gradient, LC gradient, and also the mass spec parameters and the detection mode. Either you want to choose the positive mode or negative mode. And to get a better result, you may want to include an internal standard to real time to normalize the readings of the instrument. So the major difference for the kinetic and the thermodynamic solubility is how you prepare your sample. Like we mentioned before, so the final sample contained in the kinetic solubilities, it contains 1% or a few percent of DMSO, and the thermodynamic solution solubility has not. It's only the median. So prepare the sample, and we could get a dose responsive curve. And after that, you can back calculate your water solubility of your sample. So another example I want to give here is the LC mass analysis. Here I use melatonin as an example. So it's a biomarker for sleep-related disorder. So it's kind of like very difficult to measure the level of melatonin because it's in low circulating levels. So that's why we need like mass analysis. Usually mass analysis is far more sensitive than LC mass analysis. So in this case, we developed a method for the quantitation of melatonin at 10 picogram per milliliter level by using a small amount of like 50 microliter of serum samples. So I'm not going to very detail of the assay development, but just give you a quick idea. So first, we need to do LC mass separation condition optimization to figure out retention time, to figure out the gradient and the mass spec mode. And then we need to like condition optimization on the multiple reaction monitoring. It's kind of like monitor the reaction change when the molecule first pass the quadruple mass, it chooses the mother ion, and then it goes to the collision cell. You apply energy to your molecules and then break the molecule down, and then it has a lot of secondary ions, and the top will detect the second ions you choose. So basically, the mass monitor the reaction transition. So you saw the you quantify the molecule by test the reaction transition, so which is more sensitive and is more confident way to detect your molecule. So of course we need a dose response curve of melatonin under the optimized mass condition. And we include D4 melatonin as an internal reference to normalize the sample, the instrument readings. And we also need a dose responsive curve with the melatonin as well. We need to change the method a little bit to see the dose responsive curve for the internal reference. And then we put them together, make sure they show up, they have the same pattern of the multiple reaction transition. And then after that, we're working on the serum samples and pre-spike with internal reference and the regular workup before the analysis. So next step is we are going to verify the method a little bit and then test all the samples by LC QTOF. So the last step will be doing data analysis, fitting the curve, and provide a summary of the result. So here I would like just to give you an idea of the cost estimation of the LC mass. Although you have just to see that there are 9 steps, but we don't charge all 9 steps at the beginning. So we try to break down the cost, and then we try to charge like stepwisely. So the cost will be like column cost. Yeah, sometimes we need specific columns for your assays. And then we maybe spend 1 day or 2 on the LC mass condition optimization, another 1 day or 2 to optimize the mass conditions and get a dose responsive curve in internal reference. So there's a very complicated assay validation part, which is optimal, which is optional, but also recommend, depends on your budget. For some of the analysis method is already established in published paper, so maybe we we don't need to do a assay validation again. But for some new method development, I would highly recommend we doing the method validation part. And the next slide is the rate, internal rate. So we do have internal rate, labor rate, and we also have academic rate to the other university, which is 1.5 fold of the price. And we also have industry rate, which is 1.8 times of the internal rate. And here's our major medicinal and analytical chemistry instrument rate for your reference. And here's our contact information. If you have any question about molecule design synthesis analysis, please feel free to contact me. And if you have never been to Chemical Genomics call before, I would be more than happy to give you a tour in the call. And if you have any question about other services we have, like phage display, high super screening, and SAR by NMR, feel free to contact our expert team. And thanks, and welcome to Chemical Genomics call. So I'm open to take any question you might have.
So thank you so much for your presentation. I wanted to know if you do only biological matrices, or do you do, like, for example, I do a lot of air pollution and I collect a lot of filters. And so I'd like to know the chemicals on the filters, and then I can take blood samples. And then I want to know if there's a correlation between the chemicals on the filters in the air and then also what happened in the body. And I wanted to know, do you do non-biological matrices?
Quantify
like a chemical?
Yes, we do. We do quantify both small molecule metabolites and also proteins as well. Yeah, we can we can talk, like maybe after the meeting. You can send me the reference and analyze and the system you want to look at, so I can take a look, do an evaluation usually. Yeah, and then we can go from there.
Okay, thank you.
I have a question in the chat.
Oh, okay, thank you.
Yeah,
Can you so the call has using the oh yes, and the call. Yeah, we do have like different kind of libraries. It's for either it's a diversified libraries in the course. And also we have FDA approved drug libraries, and we also have a nature product library and the semi-synthesized nature product libraries. Yeah, the cost of using the libraries, yeah, it's kind of like depends on the assay and the amount of the compound you're going to use. So I can connect you with Jingwei in our call and to figure out the screen cost of the library.
This is Jingwei.
Yeah. Oh, yeah.
Yeah. Can you hear me? Okay. So, well, like Li Fan described, we have roughly about 3 libraries with a natural compound with FDA approved drugs, and for the cost, roughly, if you have a very straightforward assay, and we can help you develop some assays as well, and the cost is roughly about 40 cents per compound for the screening. So for a very small library, for example, low-pack, the low-pack library is only 1280 compounds coming from FDA-approved drugs. And so the cost for the library screening is like $400. So it's very affordable. The reason is the School of Medicine actually put a lot of subsidiaries for the core operations. So I do recommend if you have the chance, take this advantage. All right. And of course, I'll put my emails in the chat box and feel free to contact me.
I have a quick question. So let's say I have a small peptide that block a whole protein. Can we take this peptide and get a compound that can block the same active portion of the protein?
Oh, yeah, good question. Yes, especially there are a lot of examples. If the small peptides can form an alpha helix. Yeah, that's a little bit more straightforward. There are a lot of small molecule mimics to the alpha helix.
7-mer, 12-mer, 10-mer.
Yeah. So
Okay.
we definitely can do that. Especially if you have the some binding information between your peptide and the protein.
Yeah.
So we can identify the binding part from the peptide and also the protein, and we can use that information to do to find a suitable small molecule to mimic that peptide.
Okay.
Yeah, and I think so. Rattan in our core can also provide the help on identify whether or where your small peptide binding to your protein. Yeah, that's also possible.
Okay. Good.
Thank you.